Okay, so we just finished reception. What happens to the signal next? Well, some things that I want you to understand is that there are multiple steps to these pathways. And we're going to require the phosphorylation molecules before the response. And these multiple steps in the phosphorylation cascade is going to allow for amplification of the signal. Each molecule that's phosphorylated is going to activate not just one, but several other molecules. And each of those molecules will activate even more molecules, and so on and so forth. So we get an exponential amplification. This is going to allow for a faster response. We're also going to allow for more coordination and control of these pathways. And we can build multiple responses. So we already talked about the reception part. So tyrosine kinase is going to be our example receptor here, which activates a particular G protein. We're not going to get into the details because that's beyond the AP course of super specific proteins. So once your G protein is activated, it's going to activate the first enzyme in the pathway. It's going to phosphorylate a kinase. And guess what that kinase is going to do? It's going to activate a second kinase in the pathway by phosphorylating it. These phosphates are going to be coming from an ATP that was hydrolyzed. There can be many more kinase enzymes. So kinase 2 could activate kinase 3 by phosphorylating it. And then kinase 3 could then go activate kinase 4 until we get a specific response. We may stimulate something or we're going to inhibit something. So this particular pathway is going to be for cell proliferation, cell migration and adhesion, angiogenesis. So this is a particular cancer pathway because it's going to inhibit apoptosis. Phosphatases are going to be enzymes that remove phosphate groups, and this is what's going to turn off the transduction pathway. We need to deactivate those kinases. So that's the job of phosphatases, is to remove those phosphates. Now, because our initial molecule cannot get into the cell, we need to pass on this message to the signal transduction cascade by a second messenger. And there are a couple. So these are small molecules that activate the transduction pathways. One example of this is adenyl cyclase. It's going to be activated by a G protein in the cell membrane, which will give us cyclic AMP, which is going to activate kinase 1 in the epinephrine signaling pathway. So the job of this guy is to activate the first step in the transduction. Another second messenger that plays an important role is going to be IP3. This is going to be the signal that is going to open calcium ion channels in the ER, which is going to be a gated channel. This is also going to be activated by a G protein.
Signal transduction is going to be used for those non-lipid signals. Once those signals bind to the receptor, second messengers are going to be released, which are going to activate protein kinases. The job of protein kinases is to add a phosphate group to other protein kinases, which will then activate other proteins, which activate other proteins. And it's all based upon phosphorylation. Another thing that happens is that we get amplification. This is how the message can be spread quickly and the response can be done efficiently.